Hi, everyone, and welcome to our module on DNA replication. DNA contains our genetic code. It's found in the nucleus of eukaryotic cells like human cells. It's found in the cytoplasm of prokaryotic cells like those of bacteria. And in order for a cell to divide, it needs to replicate or copy its DNA so that when the cell splits into two daughter cells, each cell can have a copy of the genetic code. I discussed DNA structure in detail in a video in the biochemistry section, but I'll just briefly review some key points here that are relevant to our discussion of DNA replication. So DNA is double-stranded, and each strand has a ribose sugar backbone as shown in this picture, made up of ribose sugar units. Those ribose sugars are connected to one another by a phosphate group, and then each ribose sugar contains a nitrogenous base, and the four bases found in DNA are adenine and thymine, guanine, and cytosine. The bases in DNA pair with one another, linking the two strands together, and adenine always pairs with thymine, as shown in this picture on the right, and guanine always pairs with cytosine. In RNA molecules, there is no thymine. Instead, there's a base called uracil, and so uracil takes the place of thymine and pairs with adenine. Guanine still pairs with cytosine in RNA. And if you look at this double strand of DNA on the right here, you'll notice that this strand here ends in a phosphate group at the top. That's called the 5' end of that strand. The other end of the strand is a hydroxyl group hanging off the ribose sugar. That's called the 3' end. On the other side, it goes in the opposite direction. The 5' end is at the bottom and the 3' end is at the top. And this is called the anti-parallel structure of DNA, and this will be important as we talk about how DNA is replicated. Nucleotides are the building blocks of DNA. DNA is a polymer made up of repeating nucleotide monomer units. At the top right side of the screen is an example of a nucleotide. It contains three pieces: a nitrogenous base, a ribose sugar, and a phosphate group. Nucleotides are synthesized as monophosphates with just one phosphate group, like this example on the top right side of the screen. They are then converted to a triphosphate form, like at the bottom, where three phosphate groups are attached. And it is this triphosphate form that is added to growing strands of DNA in DNA replication. So for example, at the bottom of the screen is a molecule of deoxyadenosine triphosphate. If this is added to a growing strand of DNA, these two phosphate groups will be cut off. The one phosphate group will remain and will link this monomer into the polymer of a growing DNA strand. Okay, so now that we've reviewed those basics of DNA structure, let's get into DNA replication. So shown on the screen is my own model of a double strand of DNA. I've got one strand going along the bottom with a 3' end here and a 5' end here. Anti-parallel to that, I have another strand at the top with the 3' end here and the 5' end here. And the nucleotides are all paired according to their nitrogenous bases. So adenosine always goes with thymidine in this model, and cytidine always goes with guanosine. The big picture of how this works is that the two strands will separate and then two new strands will be synthesized, and thus the cell will go from having one double-stranded piece of DNA to having two double-stranded pieces of DNA. So what we'll start going through now are the details of how that process occurs. And the first thing you should know is that DNA replication begins at specific sites called origins of replication, and we'll talk more about what's unique about these sites in a minute. The enzyme that separates or unwinds the double strand of DNA is called DNA helicase. It requires ATP to carry out this role. And then there are other important players in separated two strands called single-stranded binding proteins. They stabilize the single strands of DNA for DNA replication. And I've summarized these points on this slide here. The helicase enzyme unwinds and opens the double helix. It hydrolyzes ATP to carry out this role. Single-stranded binding proteins assist the helicase. They stabilize and straighten single strands of DNA. Let's talk a little bit more about the origin of replication. These origins of replication occur at specific DNA sequences that are able to attract DNA replication initiator proteins and that are also easy to unwind and open. If you look over at this drawing of DNA on the right, you'll notice that there are three bonds between guanine and cytosine bases. There are only two bonds, however, between adenine and thymine. This means it's easier to unwind DNA in places where there's lots of adenine and thymine, and indeed, that's where many origins of replication occur. These are called AT-rich sequences. They're easy to open because there are fewer bonds between adenine and thymine. Once the double helix has been opened up, the major enzyme that synthesizes new strands of DNA is called DNA polymerase, and there are a number of these enzymes. In bacteria or prokaryotes, there are four versions of DNA polymerase numbered 1 through 4. DNA polymerase 3 is the major DNA polymerase that synthesizes new strands of DNA. polymerase 1 removes RNA primers. We'll talk about RNA primers in a minute, but they're another important component of making new strands of DNA. In eukaryotic and human cells, the DNA polymerases are alpha, beta, gamma, delta, and epsilon. Polymerase gamma is located in the mitochondria, which you may know have their own DNA. The others are in the nucleus and perform various functions in synthesizing new DNA strands. So now let's talk about primers. It turns out that DNA polymerase cannot initiate replication on its own. In other words, it cannot all by itself start to build a new strand of DNA. Instead, it requires a short starter portion of DNA called a primer. This is a short nucleotide sequence, and this allows DNA polymerase to begin making a new chain. The primers are formed at the point of initiation of a new chain, and they are required by DNA polymerase in order for the enzyme to function. And there's an important distinction here between DNA polymerase and RNA polymerase, which makes RNA. RNA polymerase does not require a primer, and we'll talk more about RNA polymerase in the video on transcription. The enzyme that makes primers is appropriately named DNA primase, and what's interesting about primers is that even though they're used to synthesize DNA, they actually contain RNA. RNA is different from DNA in that it is made up of ribonucleotides, not deoxyribonucleotides. If you look at the bottom of the screen, a ribonucleotide has a hydroxyl in this position. That hydroxyl is missing in a deoxyribonucleotide, as is used in DNA. In addition, as we discussed previously, RNA has uracil instead of the base thymine. Eventually, these RNA primers will be removed and replaced with DNA, but they are used to help DNA polymerase get started in the process of replicating DNA. So here's a simplified picture that you can keep in your mind to understand how this process works. The primer binds here at the 3' end of one of the strands at the origin of replication. This will allow DNA polymerase to then begin adding nucleotides so that it can replicate this upper strand here. So that brings me to an important concept called the directionality of DNA synthesis. In order to understand this, let's imagine that these 4 nucleotides I've placed on the screen are linked together in a molecule of DNA. So we've got a strand of DNA with a C, A, T, and G sequence. And let's imagine that DNA polymerase would like to add an adenosine to this DNA strand. Well, nucleotides are always added in a triphosphate form. So this is adenosine triphosphate, and it's ready to be added to this strand. What DNA polymerase will do is break this phosphate bond here, and that will create the energy to add this nucleotide to this strand of DNA. So the way you can understand the directionality of DNA synthesis is to appreciate that all the energy comes from right here. In other words, it's like there's a little bit of heat around this phosphate bond that is going to seal this phosphate bond onto the growing strand of DNA. Well, the only place the phosphate bond goes is down here at the 3' end. It would not link to this phosphate group here at the 5' end. So what this means is that DNA polymerase will always add nucleotides to the 3' end because that is where the phosphate group fits, and the phosphate group is the source of energy used to add new nucleotides to a growing strand of DNA. So if DNA polymerase were to add that A to our growing strand, we would go from 4 nucleotides to 5 nucleotides, and the new one would be added down at the bottom where the 3' end of the strand was. So the key point here that you want to remember is that DNA replication or DNA synthesis always occurs in the 5' to 3' direction, or another way to look at this is that nucleotides are always added to the 3' end of the growing strand, and that's because that's where the phosphate group fits, and the phosphate group is a source of energy. So if we go back to our replication fork here, the primer has been put down here. This is the 3' end of the template that we're using to make new DNA, which means that the primer has to be anti-parallel. So this is the 5' end of the primer, and this is the 3' end of the primer. And as we discussed, nucleotides are always added to the 3' end. So what's going to happen here is DNA polymerase is going to add another base, another base, and another base to grow this strand of DNA. Now that works fine for the top strand, but in the bottom strand, the orientation is opposite. So the 5' end of the primer is over here and the 3' end is over here. This means that in the case of the bottom strand, the primer has to bind closer to the branch in the double strand of DNA, and it's got to add molecules in this direction. So therefore, DNA polymerase on the bottom strand will add molecules like this to synthesize the bottom strand. Now let's talk about what happens as DNA continues to be unwound and the replication fork widens. For the top strand, this creates no significant problems for DNA polymerase. Nucleotides can continue to be laid down as the replication fork widens. For the bottom strand, however, there's a problem. DNA polymerase has already used the primer that was laid down to synthesize a new molecule all the way to the end of the old molecule. What this means is a new primer must be laid down, which DNA polymerase can use to synthesize DNA all the way up to the first primer, and thus the DNA synthesis in the strand on the bottom is going to be occurring in pieces, whereas the DNA strand synthesis at the top is going to be continuous, and that's because of the different 3' and 5' orientations of the top and the bottom strands. And here's a picture from the web just to pull all this together. The top strand is called the leading strand. It is oriented from 3' to 5', and thus it only requires a single primer, and then DNA polymerase can continue to synthesize a new strand as the replication fork widens. The bottom strand is called the lagging strand. It is oriented from 5' to 3'. It requires multiple primers and will synthesize DNA in fragments, which are called Okazaki fragments. They will eventually be linked together in a continuous strand of DNA. Let's talk about how primers are removed. As we saw in the last slide, for the lagging strand, there are a number of primers. The Okazaki fragments are individually synthesized until a primer is reached, and then the RNA primer is removed and replaced with DNA. In prokaryotes, this role is carried out by the enzyme DNA polymerase I. In eukaryotes, it's carried out by DNA polymerase delta. Once the primer is removed and replaced with DNA, DNA ligase joins Okazaki fragments together in the lagging strand. This enzyme creates phosphodiester bonds. These are these phosphate bonds in the backbone of DNA, and when they are created, that will link together the Okazaki fragments. There's one more player in this process we need to discuss, and that is the enzyme topoisomerase. This enzyme prevents DNA tangling. As DNA is being unwound, it can get knotted up and tangled, as shown at the bottom left side of the screen. Topoisomerase enzymes relieve that knotting and tension and tangling in DNA. The way they do this is by breaking the DNA and then resealing it to relieve the tensions and twists. Topoisomerase I is an enzyme that can break single strands of DNA and then reseal, and topoisomerase II is an enzyme that can break double strands and then reseal. And there are a number of clinical correlations with the topoisomerase enzymes. The quinolone class of antibiotics inhibits prokaryotic topoisomerase enzymes. This leads to breaks in DNA strands that cannot be resealed, and thus DNA damage occurs, and that's how these antibiotics kill bacteria. There are also a number of chemotherapy agents that target eukaryotic topoisomerase enzymes. For example, adiposide and tenofovir, irinotecan and topotecan, and the anthracyclines. All of these have activity that inhibit topoisomerase enzymes, and this disrupts cells that are rapidly dividing, like cancer cells. Let me make a couple general key points about DNA replication. These are the kind of things they like to ask you on exams. So leading strand DNA replication is continuous, as I showed before, but lagging strand replication is discontinuous. That involves the Okazaki fragments and DNA ligase enzyme to link those Okazaki fragments together. DNA replication is referred to as semi-conservative. The reason it's called this is because the new DNA strand contains one old and one new strand. So for example, if we start with a double-stranded piece of DNA, the strands will separate and we'll get two new strands that each have one of the old strands, and that is termed semi-conservative replication. Now that we've discussed DNA replication, let's talk about proofreading. Sometimes when a new strand of DNA is being synthesized, the wrong nucleotide can be inserted. It turns out that DNA polymerase has the ability to correct these errors in some cases. As we talked about before, new strands are always synthesized by DNA in the 5' to 3' direction. If the wrong nucleotide is added, DNA polymerase can move backwards. In other words, it can go in the 3' to 5' direction in order to correct the error. When DNA polymerase does this, it has enzymatic activity called exonuclease activity, which allows it to remove the incorrect nucleotide. So you will often see it written in your textbooks that DNA polymerase has 3' to 5' exonuclease activity. This means it can move backwards along the new strand to fix an error, and this significantly reduces the error rate during DNA replication. Just to make this point very clear, let's go back to our drawing of a replication fork. We have the leading strand on the top with the 3' end here and the 5' end here. DNA polymerase is always going to synthesize the new strand in the 5' to 3' direction. So in the new strand, this is the 5' end, and over here we have the 3' end. If the wrong base is inserted, as I've shown here, with adenosine being paired with cytidine, DNA polymerase can move in the reverse direction. In other words, it can move in the 3' to 5' direction along the new strand and remove that incorrect base, and this is its role in proofreading new strands of DNA for errors. The last enzyme I'll discuss in this module related to DNA replication is telomerase. Telomeres are nucleotides found at the end of chromosomes. They often contain repeating sequences of TTAGG. And the problem at the end of a chromosome is that there's no place for an RNA primer on the lagging strand. This is especially true in eukaryotic cells, which have non-circular DNA. So what the telomerase enzyme does is recognize telomere sequences and add these sequences to new DNA strands so that nucleotides are not lost as each DNA strand is replicated. Telomerase contains RNA molecules that are used as a template to synthesize the DNA at the end of chromosomes, the telomere DNA. This is referred to as having enzymatic activity of an RNA-dependent DNA polymerase enzyme, and this is similar in many ways to the reverse transcriptase enzyme, which can be found in viruses like HIV. This enzyme takes a piece of RNA and uses it as a template to synthesize a piece of DNA, and that DNA will replace the end of the telomere, which would otherwise be lost if not for the telomerase enzyme. So here's a picture to make this more clear. This is the telomere. This is the end of the chromosome here. You can see that it has TTAGG sequences near its 3' end. So this is where telomerase will come in and bind. Telomerase has an RNA template as part of the enzyme, which can bind to the telomere and then begin to extend the end of the telomere in this direction. Once it's extended the 3' end of DNA, DNA polymerase can come in and complete synthesis of the lagging strand in this direction. And this avoids loss of genetic material with duplication. If you didn't have this end of the DNA strand would not be replicated and it would be lost in each copy made of DNA. You can imagine that in cells with absent or low levels of the telomerase enzyme, after each cell division, genetic material will be lost and eventually the cell will no longer be able to divide. For some cells, this is a big problem because they need to divide indefinitely, and so those cells are where you find lots of the telomerase enzyme. So for example, in hematopoietic stem cells, the telomerase enzyme is present and it allows for controlled indefinite replication. You can also find higher levels of the telomerase enzyme in cells that divide indefinitely for other reasons, like cells of the epidermis, hair follicles, or certain cells in the intestinal mucosa. In addition, the telomerase enzyme is implicated and may play a role in many cancers. This allows the cancer cells to become immortal, and in fact, there is research underway to try and inhibit telomerase enzymes in order to treat certain cancers. And that concludes our video on DNA replication.